Let's talk about our genes. These genes.
If you had a hole in your favorite pair, you could fix it by patching it up with new fabric. If you didn't, you might have a little problem. Now, let's talk about your other gene, the DNA that encodes all of the proteins in your body. Unfortunately, defection in our genes can cause life-threatening diseases, and these are not curable by traditional methods like taking readily available medicines such as pills and over-the-counter medications. And this is the point which gene therapy gives hope in the medical field. He is William French Anderson, the father of gene therapy. In 1990, a 4-year-old child with ADA deficiency received the first gene therapy. Want to know more how gene therapy started? In 2015, researchers were able to restore basic hearing in genetically deaf mice using gene therapy. They used an improved gene therapy vector developed at the Massachusetts Eye and Ear.
Our strategy was to take a viral vector, remove the viral gene so that it doesn't make anyone sick, and replace those with the correct DNA sequence for TMC.
Human gene therapy was created in 1971, the same year that the first recombinant DNA experiments were planned that involves inserting foreign DNA into the patient's tissue in order to cure diseases. While gene therapy is undoubtedly promising, but it is not favorable in medical field to be studied. There are several approaches to gene therapy. Replacement of mutated gene that causes disease with a healthy copy of the gene, inactivation of a mutated gene that is functioning improperly, introduction a new gene into the body to help fight a disease. A gene cannot be directly inserted into a human gene or cell, but it can be inserted in another gene using a carrier or vector. An example of this is virus that involves in a way of transporting their genes to human cells in pathogenic manner by infecting the host cell. Gene therapy can be given through ex vivo or in vivo methods. In vivo gene therapy is being done inside the human body by using injection. New genes are inserted directly into your body, while ex vivo gene therapy is done outside the human body by culturing cells in a test tube or a culture dish and placing them back into your body.
First form is called somatic gene therapy, which involves the manipulation of genes in cells that will be helpful to the patient but not inherited to the next generation. The other one is called germ-line gene therapy, which involves the genetic modification of germ cells that will pass the change onto the next generation. Stem cells has the ability to self-renew or multiply while maintaining the potential to establish into another type of cell. It is probably to become cell of blood, heart, bones, skin, muscle, and brain. Stem cells is derived from these two sources, which are embryonic and somatic stem cells. The embryonic stem cells are derived from a 4 or 5-day-old human embryo that is in the blastocyst phase of development. The somatic stem cells are cells that exist throughout the body after embryonic development and can divide that enables them to generate a range of cell types from the originating organ or regenerate the entire original organ. Above all this information, the pros and cons of gene therapy should be considered. It benefits humankind as it provides options. In cases where no other treatments have been effective, gene therapy may be able to cure a patient. It only has to be given one time or applied as a single dose. Comparatively speaking, this is appealing to certain other therapies that seek for repeated doses. Lastly, in some cases, symptoms can be permanently eliminated with gene therapy.
On the other hand, it also gives disadvantages in some aspects. Gene therapy can be quite expensive, which makes it unavailable to some people who can't afford it. Another is that gene therapy is experimental and there are still many things we don't understand about gene therapy despite its recent development. To conclude, gene therapy raises ethical concerns for certain people. One such concern is who will determine the good and bad applications of gene therapy and which traits are normal and considered as disorder. Advances in gene therapy research allows to treat and hopefully someday avoid rare and debilitating diseases.